Hello. Thank you, Mac, for organizing this amazing symposium. So today I'll be talking about dynamical neuromodulation of functional network states. So I will start showing some evidence from real data about how really one molecule could be, I'll go like this, how really one molecule can modulate the occupancy of different functional network states. I will then briefly introduce a possible mechanistic hypothesis and how we can use computational modeling to test this possible hypothesis, so to explain why we may be observing these things from the data. So we all know about bold activity. I will go quickly on this, but so if we take two regions of interest, be they voxels, large areas, I'm not going to discuss on parcellations, but if we take two areas and if we measure their any type of temporal dependency, again, it can be correlation or other different measures, we can infer what is called functional connectivity. But then if we consider a set of different networks, we can talk about a set of different areas, we can talk about a network. So there can be distant areas that for some reason seem to be operating together. And this, in a functional connectivity matrix, appears as a square, meaning that those areas are all correlated together. But functional connectivity is dynamic, as you all know, so these patterns of connectivity or squares that we detect in the connectivity matrix that we can apply graph theory to seem to be changing all the time. So this means that there are different functional subsystems that are constantly being formed over time. And if we really manage to go into faster temporal scales, even with the fMRI signals, so this is a measure that considers instantaneous, let's say, functional connectivity by focusing on the synchrony of the bold phase at each instant of time, we can see that there is a constant evolution of functional patterns. Now, we can use different methods to see if these functional patterns reveal different functional networks and if these networks are recurrent. So I think over the last decade, we've seen some consistency on these so-called resting state networks that appear to be recurrent patterns of functional connectivity that constantly reoccur over time. This is using a k-means clustering algorithm applied using a method called LIDA that I've proposed in 2017, and we've recently had 2 papers using this method. But basically, the idea is, if we have this repertoire of functional network states, and we can represent it in a low-resolution manifold like Mac nicely introduced. So each cross there represents represents the centroid that represents each of the functional network states. And in this sense, we can look at a single session, fMRI session, and see it as trajectories within this low-resolution manifold. So this flows on a manifold seem to be an hypothetical scenario for how brain activity or how dynamic functional connectivity emerges. But importantly, these functional network states reoccur in different sessions, in different subjects. And the thing is, what modulates this dynamical exploration of the repertoire of functional network states during rest? And more importantly, that I've been thinking is, does network occupancy, like the time you spend close to the different centroids, relate to behavior? So to address this question, we took a data set that is on psilocybin, but so psilocybin is the active ingredient in magic mushrooms. I'm not in the psychedelic research. I just look at this from a very objective perspective where you have a resting state scan, okay? You have a scan. This is a fMRI session one time, and then inside the scanner, the patients are injected with the purified version, so it's psilocybin, it's the active ingredient, a psychoactive molecule, let's say, and then we continue recording the fMRI signal. So the subject doesn't move. And then there is another session where the participants go again into the scanner and they are injected with a placebo. They don't know which session they are injected with psilocybin or a placebo. This is an interesting data set. We're doing secondary use of this data to study how can a single molecule that acts on the serotonergic system modulate this dynamical exploration of the repertoire of brain networks. So importantly, using this clustering analysis that I showed before on the different states, we can, for each session, we can quantify how close we are to each of the different. These were the 7 centroids that I showed before. So it means when there's an epoch here in blue, it means that we were close to a certain network configuration, and so on. And with this, we can calculate the probability of occurrence of each state, which I will call network occupancy. And we can also calculate the duration, the switching probabilities, and these things. So in this data set, what we found was that when they were injected with placebo, so here is the different states, and here is the probability of occurrence or the occupancy of each of the networks. So this is before and after injection of placebo. I will start with the placebo because then it's more illustrative. So we see that before and after the injection, the probability of occurrence of each of the states remains, there's no significant difference across subjects or across sessions, both. Then if we, sorry, it's here. Then before injecting psilocybin in the resting state, also very nicely we see that the probability of occurrence of each of the states remains constant as well. But now when we inject psilocybin, it's here, what we find is that the occupancy of a specific network state, so the trajectories to closer to that centroid, are significantly reduced. They are there around 15% of the time in all non-psychedelic conditions, and then they are reduced to around 3% of the time after psilocybin was injected. So this is particularly interesting because, so if we look again at this cloud or this low resolution manifold, that in this case, which I tried first to represent this on a landscape, but then we thought it's better represented in a cloud because actually it is easier to see in 3 dimensions. But of course, this is a multi-dimensional space. This is reduced to 2. So this is the probability, so the time spent in each of, closer to each of the centroids before any injection. Then we inject the substance, either psilocybin or placebo, and we'll see the clouds again. And now there's a question. So here is a game. You have to guess if psilocybin was the red or the blue injection. So the thing is, just by looking at this cloud and seeing, it is almost visually striking, maybe because I know already, that okay, that the red part of the cloud, so there is a region of this low resolution manifold that is significantly less visited under the influence of psilocybin. Now, not only we found this, but also the subjects after the scan, they were asked to rate their subjective feeling of the psychedelic experience. And consistently, all of them, of course, rated much higher the psychedelic experience. So in the vertical axis is this rating, and the red are the psilocybin experience. So they always rated higher. The red dots are all high, and in blue are low ratings, of course, of the placebo effect. So they could feel the psychedelic experience. And here we correlate it to the network occupancy of each of the states. And we see that for all subjects, consistently, the feeling of a psychedelic experience was related to a decrease in the probability of occurrence of this frontoparietal system. And importantly, this frontoparietal system has been previously related to executive control. I should have added some references, but it's a network. It's still unclear what exactly it does, but what we find here is that, again, a molecule somehow, by some magic, modulates the occupancy of the different networks, and once you are there, apparently the people describe this feeling of ego dissolution or unconstrained cognition. So now a bit on mechanistic hypothesis. How can a network dynamics be controlled by a neuromodulator? So it's a psychoactive substance, so there are many. Everywhere, every day we have not only psilocybin, caffeine, valerian, there are many other molecules that are psychoactive and then change the way we behave. So as we've seen some previous introductions, the thing is that there are in our brain, we have receptor maps. And these molecules, they bind to specific receptors. In this case, psilocybin and other psychoactive molecules bind to serotonergic receptors. And so here I should have put the reference. This is the work by Béliveau in Journal of Neuroscience 2017 in collaboration with Gita Knudsen. She gave a keynote last year in Singapore. So it's there are new imaging techniques from PET that allow us to look at different receptor maps in the brain. And what this is revealing is, for example, these are different types, subtypes of serotonin receptors. So it's the 5-HTT, 5-HT2A, 5-HT1A. There are different types. And we know, for example, that psilocybin binds preferentially to the serotonin 5-HT2A receptor. So the idea is, can we integrate this receptor-specific neuromodulation in brain network models? So, as Michael introduced, so the idea of these brain network models that were used to simulate resting state activity are networks, so that simulate the behavior of coupled neuronal masses. So each node in this network represents a large population, like the collective activity of a large population of neurons. This is described by equations, but as we've seen before, there are equations in this model that describe the gain of each brain area. That is, how much, given the input current that that brain area receives, how much it increases in terms of firing rate. Okay? So the idea is to heterogeneously manipulate node dynamics according to different receptor density maps and see which best fits brain activity in the psychedelic state. So this work I will be showing was actually not using the psilocybin dataset, but the LSD dataset. But the idea is that both these molecules bind preferentially, of course, because they bind to different. We still don't fully understand this receptor binding mechanisms, but they both bind preferentially to 5-HT2A. So very briefly, here we focused at the time. We were just focusing on fitting the temporal similarities of functional connectivity dynamics. So not specifically brain network, but trying to fit the functional connectivity dynamics, which is a matrix of time versus time correlation between Windows averaged functional connectivity matrices. So the idea is first run the model with resting state parameters adjusted to the placebo fMRI session. Then, so here is, we show here, how we obtain the best fitting, the best global coupling parameter. Then, we change the excitatory gain modulation, which is we change the slope of the curve of the input-output function that scales the degree of firing rate of the excitatory population. And we see that as we increase the gain modulation in specific brain areas, we obtain a better fit. So when we minimize this fitting, it means the fitting is better to the LSD data set compared to the placebo. Also, not only we reshuffled the receptor map and I showed that the fitting is worse, but we also tested with different receptor maps, all the other receptor maps, or even a uniform receptor map. And here in this paper, we show that using the receptor map where LSD preferentially binds, we are able to obtain the best fitting of the computational model. So now that I'm past time, it's just 2 slides, but overall, I'd like just that you take home this message that neuromodulators can actually control the dynamical properties of functional network states, so we can see this from the data. And importantly, understanding how molecular neuromodulation acts on macroscopic functional networks. So because there has been a lot of research understanding how it works neurophysiologically and at single neuron levels or neural populations in vitro, but understanding how it sculpts brain activity at the macroscopic level may shed light on the missing link between pharmacodynamics and brain function. So the idea is that this could propose new theories for psychiatry, new therapies as well. So if we can understand which are the pharmacs that could be prescribed. So these are the latest results that I've found where the occupancy of functional networks using this method relates not only to the psychedelic experience, but also to cognitive performance, also to vulnerability to depression. And now we have some work also being ongoing work that was presented yesterday at the poster, where we show that the occupancy of these different networks also relate to obsessive-compulsive behavior. So overall, I'd like to thank all my network of collaborators. I am based in Portugal, affiliated at Nuno Sousa Lab in the University of Minho. I spend most of the time in the lab of Noam Shamesh in Portugal. So these are the posters that were presented here and will still be presented at OHBM. And I'd like to thank the funding agency in Portugal for supporting me, my children, and the third one on the way. And thank you everyone for being here. Thank you. Okay.